Good afternoon, friends. Today we will discuss one important staining technique in microbiology that is called negative staining. The purpose of negative staining is to observe the morphology and arrangement of bacterial cells. As we know that bacteria, they are very small microscopic organism. Even if we want to observe them, we need to color them, and the staining is a coloring method, but this negative staining is an indirect staining method. This indirect staining method will help us to observe the morphology as well as their arrangement. This negative staining is also used in electron microscopy during the preparation of a specimen, and in negative staining uh, it is used to stain the cells that cannot be heat fixed. In generally in most of the staining techniques you need to heat fix the cells, but in this staining method you need not to heat fix the cells. The principle behind negative staining. This negative staining requires use of an acidic stain such as India ink or nigrosin. Remember, whatever the staining technique is used in microbiology, certain type of stains are used, like acidic stains or basic stains. Basic stains are used in simple staining, monochrome staining or gram staining. In negative staining, acidic stains are used. Means the stain possesses negative charge, and we know that bacterial cell wall has already a negative charge. Bacterial cell surface has already a negative charge. So what will happen? These stains like India ink or nigrosin will not stain the bacterial cells, because charge on bacterial cells and charge on these stains is same. So there will be repulsion between the stain, that is nigrosin or India ink, and the bacterial cell surface, because both of them have like charges, negative charges. So any of these stain will not stain the bacterial cells, or bacterial cells will repel the stain. Means whatever is the color of the nigrosin or India ink, this color will not bind to rather the stains itself will not bind to bacterial cell surface. So what will happen at the end? The cells will be unstained and are easily observable against a dark background. The cells will be unstained, remember. Therefore, this method is called a negative staining. Although we are using the stains, but these stains are not actually staining the bacterial cells, because they have similar charges. Charge on the stain and bacterial cell surface is similar. So cells will remain unstained, and these stain will stain the background rather than microbial cells. So we will find that background will be a dark background and cells will be unstained. Let us see the procedure behind this negative staining. Place a drop of nigrosin close to one end of the clean slide. In most of the staining technique, what we do, we take drop of drop of the stain at the center. But in this method, what is done? a drop of nigrosin or India ink is taken at one end of the clean slide, and under aseptic conditions a loopful of the inoculum from bacterial culture is placed in the drop of nigrosin and mixed. Means where you place the nigrosin, a drop of nigrosin, at the same place take a loopful of the culture and mix it with nigrosin. So what is you are doing initially? You are taking a drop of nigrosin and then bacterial culture, mixing it with the nigrosin at one end of the slide, and place a slide against the drop of suspended organism at 45 angle. So place a slide against the drop of suspended organism at 45-degree angle and allow the drop to spread along the edge of the applied cell. Or you can take another glass slide, hold the slide at 45 degrees Celsius and prepare a very thin smear by holding the slide at 45-degree angle. So in this way, this such type of thin film is prepared on the slide. This smear should be thin and then slide should be air dried. Remember, in this step, in this negative staining technique, the cell is not heat fixed. In simple staining, in gram staining, we firstly heat fix the cells and then place a drop of stain. But here the cells are not heat fixed, they are air dried, and then after air drying, observe the slide under oil immersion objective. This is the procedure behind negative staining. The observation, the bacterial cells will be observed as transparent against dark background. This is the observation. Bacterial cells will be transparent and background will be darker. It is because the stain, that is India ink or nigrosin, they have negative charge, and as well as charge on the bacterial cell wall is also negative. So there will be repulsion which will not stain the bacterial cell. The stain will stain the background rather than bacterial cells, and cells will become transparent. Cells will be unstained, and therefore this method is called a negative staining.